the Drug Discovery Lab, MCDB 1171. Amir Podoswa will be presenting his talk. And Amir, when you're ready, you can go ahead and share your slides.
Hello, everyone. My name is Amir Podoswa. I am a student at the Drug Discovery Lab, and today I will be presenting on our discovery of novel antimicrobial compounds to address antibiotic resistance. Here's an outline of what we're going to be looking at today. And I think first and foremost, it's important that we understand what is an antibiotic-resistant infection. And to do so, we have to understand what is an antibiotic. So an antibiotic is any drug that stops the spread of bacterial infections. It does so by either killing the cell or stopping it from replicating. And much so if you were targeting any other living being, you have to target its vitals, right? So an antibiotic will target the cell wall, the capability of the cell to synthesize DNA, RNA, or proteins. And each antibiotic is categorized in different classes, right? So each class of antibiotic targets one specific type of cell machinery. And as we all know, we have been using these for a relatively long time now, and they have been reliable. So how exactly does an antibiotic-resistant infection develops? So bacteria is constantly replicating, and oftentimes they mutate, and these mutations can lead to them being immune to certain antibiotics. So when we have a population, as you can see here, of bacteria, and within it there are a couple of bacteria that are already resistant to an antibiotic, and we expose this population to an antibiotic, what it allows is it creates an environment for the resistant population to outcompete the rest of the bacteria and reproduce and create a abundant resistant population. It is important to know that when a bacteria develops a resistance, it will also develop a resistance to almost every antibiotic within its class. So for example, penicillin and cephalosporins, they both target the bacterial cell wall. And if a bacteria develops a resistance to penicillin, it is all right to assume that it might have developed a resistance to cephalosporins as well. So doctors, usually when presented with an antibacterial resistant infection, will prescribe you a different type of antibiotic from a different class in order to circumvent the issue. And here I pictured a doctor that seems to be lightly nervous, because every doctor, every time that he treats or she treats a bacterial infection, they become slightly nervous, because what if they become resistant to other classes of antibiotics? So, which leads us to the problem. What exactly is the problem? Well, the problem is that we have not discovered any new classes of antibiotics since the 1980s. And this is for a couple of reasons. For one, it is not really economically feasible for pharmaceutical companies to develop all these new drugs. And also, most of the antibiotics that were easy to discover, they have already been discovered, right? So we are now present in a time that we would call the discovery void, where we haven't discovered anything new. And all at the same time, infections have continued to develop resistance to multiple classes of antibiotics, which leads us to today, right? So today in the United States, it's expected that there are around 35,000 deaths a year from antimicrobial resistant infections. Worldwide, the number is estimated to be about around 750,000 per year. And I am sure a lot of you have heard of MRSA, which is multiple resistant Staphylococcus aureus, and MDR-TB, which is multiple drug resistant tuberculosis. Both of these infections have infected millions of people worldwide, and they are probably responsible for hundreds of thousands of deaths. And as we can see in this infographic, a normal Staphylococcus aureus infection that has a high mortality rate of 11.5%. Once that infection mutates to become drug resistant, the mortality rate more than doubles, and the price to treat the infection doubles as well. And if you think this is bad, it is actually getting much worse. And the reason why, it is our overuse of antibiotics, right? From our crops to our livestock to our fish farms, even our industrial products, they have all overused antibiotics, as well as doctors overprescribing antibiotics. And we are essentially creating an environment suitable for the bacteria that is resistant to proliferate. And it is important to understand that we are not actually causing the bacteria to mutate faster. What we are causing is we are providing it an environment for it to succeed, right? This will lead that if by 2050 we continue on the same path, antimicrobial resistant infections are predicted to become the #1 cause of death in the United States, with over 10 million deaths a year. That is more than cancer and diabetes combined, which is fairly substantial, right? Which leads us to our research, right? So our research objectives are to identify potential compounds that exhibit antibacterial activity, right? So this semester, we performed compound screens with over 1,200 compounds that were sent to us from the National Cancer Institute, and as well as some students pursued independent compounds that they had previously perceived to have antibiotic or antimicrobial activity, right? And what this allows us to do is that this allows us to form hypotheses about other compounds based on their known mechanisms of action, right? Photo here is student Madeline Barr from one of our labs. The organism we use to test this is Salmonella typhimurium. Salmonella typhimurium is a gram-negative bacteria that is rod-shaped, and it is commonly responsible for food poisoning. But it is a very good bacteria to work with because its incubation period is very low, which allows us to just dole out the results constantly. And it also serves as a model for typhoid fever. Its sister bacteria, Salmonella typhi, is responsible for typhoid fever, which is a very deadly disease that has infected hundreds of thousands and killed hundreds of thousands worldwide. And this bacteria actually causes the same symptoms of typhoid fever in mice. So we can make assertions and try to understand the disease better. And another unique aspect of our experiment is that all these bacteria were cultured in something called M9 minimal media. And M9 minimal media is a media designed to replicate the conditions within a human being in which the bacteria is present. So the way we tested this is, in essence, we would expose the bacteria in a 96-volt plate to one of the compounds. Then we would culture the bacteria for 24 hours, and we would test it with a spectrophotometer to measure the concentration of bacteria. And then we compare the concentration of bacteria with a negative control, and we would see if the bacteria actually grew or it died or it didn't grow, right? So far we have talked about what an antibiotic-resistant infection is, how the problem is getting worse, and the objective of our research. And right now we're going to jump onto our results, and I would like to highlight that from this year's compound screen from the National Cancer Institute, we actually were able to identify 8 potential compounds that could be used as for further research into antibiotics. However, I'm going to take this time to highlight some of the independent research that has also yielded some positive results. So we begin with Emu-8 cream. Right here on the top right corner, there is the number of the posters, and I highly encourage you guys to go check them out. The research is very interesting. And right at the bottom is the names of the people responsible for this research. So this group of researchers, one of the moms of one of the researchers, she swore for the antibiotic capabilities of Emu-8 cream. So the group decided to test it. And while it is difficult to test cream, they found a list of active ingredients, and they found this active ingredient called photosphingosine. And they decided to test it for antimicrobial capabilities. And as we can see here in this graph, it actually didn't work. So they were left wondering, well, what else could cause it to have antimicrobial capabilities? And they noticed that there was silver in it. Luckily, my group, we were actually testing silver nitrate, right? And our idea for choosing silver nitrate was we wanted to have a highly electrolytic compound that would dissociate in the media and would create a high concentration of silver ions that would theoretically kill all the bacteria, right? And we started having some issues because, and they are pictured here, a precipitate would form whenever we expose silver nitrate to the media. And we were left wondering, well, what if we could test silver and have it not react with the media? And luckily for us, there was another group that were actually testing silver nanoparticles. And a silver nanoparticle would be the same as a silver ion, except it doesn't have a charge. So it will not interact with the media. And as we can see here, they got very clear results in their dilutions, demonstrating the clear capabilities of silver nanoparticles as an antibiotic. Well, not as, as an antibiotic. I would also like to highlight another research that was conducted on epigallocatechin gallate. And epigallocatechin gallate, it is, or EGCG, is a compound commonly found in green tea. And this compound has been theorized to have many health benefits before. And this group of researchers decided to test it for its antimicrobial capabilities. And what they actually found was very interesting, because at higher concentrations, it demonstrated less antibiotic capabilities than at low concentrations. Nevertheless, it showed the antimicrobial capabilities, right? And having a negative slope is something that is very interesting. I highly recommend everyone check out the poster D20. It is, in my opinion, very well done, and you could learn more about this. So where do we go from there, right? So from there, all of the compounds that we tested, a list of the ones that of the ones that worked is compiled by our very own Pamela Harvey, and it is shared with the principal investigator and sponsor of research, Corey Detweiler. If one of the compounds looks particularly interesting, her lab may pursue in the future research, which would involve testing the compound in different types of media, conditions, and animals. And then that research is then published, and it is free to use by pharmaceutical companies, which means that we have essentially did the preclinical trials test for them. We would like to thank the principal investigator and sponsor of research, Corey Detweiler. We'd also like to thank the Howard Hughes Medical Institute, the Department of MCDB, the Biological Sciences Initiative for the funding resources. And lastly, we would like to thank Dr. Pamela Harvey for helping us through this process, and also our great TAs that were very much involved. Thank you. I will now be taking any questions.
I just realized real medical research doctors are presenting.
Okay, we can take any questions that you have. It probably would be useful, given the number of people here, if you could raise your hand, and then you'll pop up to the top of our screen. We'll also look at the pictures, or sorry, the comments section in the chat. Amir, I have a question. For the two that you presented, I assume that you chose them because you felt that they were, you know, the most interesting results, but also potentially the most promising. If you were to continue in the lab for another semester, what would you do? What would you choose?
How would I choose to continue with our research?
Yeah.
Well, definitely I would choose to test it out in a different media, right? We got to a point where we were having very promising results, but we couldn't really, if I can show you here, right? They weren't really showing up as statistical hits. So I was very frustrated because I saw that this could work. So if I could just keep trying it on different conditions, and I'm sure it will be way better.
Yeah, statistics are hard, right? Because they're actually, it looks qualitatively like it's working as good, as well, or better than ampicillin, which is really promising, but it's true, we have to stick with statistics.
Exactly.
Joe, you have a question?
Yes, hi, Amir. My question is, when you put that silver in there and you get certain results, let's say you put in maybe cyanide, for example, and it kills all the bacteria. How do you know that this chemical you're finding is not going to be killing the host or the person that you give it to?
Well, that's a great question. That's actually part of our further research, where we developed something that's called a therapeutic index, when we determine at what concentrations could this compound be toxic to a bacterial cells, and at what concentrations it would be toxic to, let's say, a human cell, right? We have a lot of previous research and a lot of previous literature that I have read about silver nitrate being considerably more toxic at lower concentrations to bacterial cells than they are to human cells. But normally that would be found out in a further stage of research.
We have a question in the chat. Emma is asking, Do you know why the green tea compound worked better in smaller amounts?
Well, there are a couple of reasons why that could happen, right? Some compounds work better at lower concentrations, but it could also have a dual effect where it can act as a kind of a nutrient. I am not completely prepared to answer this, but if you read their poster, I am sure they go into it a little bit more deeply, and it is very interesting in my opinion.
Yeah, that's very true. I think for a lot of the compounds that are derived from food in the class species, what happens is it's kind of a balancing act between a compound serving as a nutrient to a cell and also serving as an antimicrobial, and it's sometimes really hard to tease those things apart. There's also something called a U-shaped toxicity curve that some compounds exhibit, like endocrine disruptors.